So how can we actually go about detecting and answering these kinds of questions with the tools that we have? Well, first off, a lot of this was not really possible until the advent of single-cell sequencing. So just as a brief primer on this, you have this tissue, which I represent here as a fruit bowl, and different cells. This is a very popular way of doing this kind of explanation. Have each cell represent a different kind of fruit. And if you do bulk RNA sequencing, which is what we used to do in the past, you essentially create a smoothie and shred up all of your cells into this slurry that's an average signal, transcriptomic signal from your sample. As you can imagine, you can't really detect signals happening between cell types or different kinds of cells if you're just getting a single signal. So using single-cell transcriptomics, we can actually isolate the transcripts from each one of these cells by barcoding them. And then we still have the information of which transcript came from which cell, and we can then look at the communication occurring between each cell type. So how does this data? How does this actually work? Well, first off, the spatial transcriptomics is becoming more and more popular, as I'm sure many of you have seen. Ever since around 2018, there's been exponential growth. This is mainly due to the increased number of technologies that can sequence more and more cells for cheaper and cheaper costs. And some of the most popular ones include the Chromium controller, which I'll briefly go over how it works due to its popularity and its availability in many of the institutions around here as well. So this is using a microfluidics platform. So the way this works is you have these barcoded gel beads, and on each one of these gel beads, you have these oligonucleotides with a partial Illumina sequence, along with a barcode. So this will determine which transcripts came from which cells. All of these kind of tags have that same barcode, but then that's also attached to a unique molecular identifier in order to control for how many reads are being attributed to each transcript. And lastly, a poly-DT tail, which can bind to the poly-A tail of the transcripts in order to attach them to this bead. And so cells are kind of fed through along with the beads, and they're captured in these kind of bubbles surrounded by oil. And in theory, there should be about one cell for each one of these beads. You can then use reverse transcription in order to get the cDNA of the transcript along with these barcodes. And then you can pull everything together by removing the oil and then do normal kind of Illumina sequencing in order to sequence the entire transcript along with the barcode. So now you know which transcripts came from which cells. And so here's an example that won't work because I'm using a different program. That's all right. It looks really cool. Trust me on that. So the typical workflow will generally end up with a map that looks like this, where each one of these dots represents a single cell, and they're kind of similar to each other within gene expression space. The way you get this is by having all those transcripts as output from the Illumina machine in like a BCL file. You usually use a program like Cell Ranger or another kind of alignment tool in order to get the barcodes representing each cell in this matrix, along with the number of reads that were found in that cell aligned with a particular gene. So you'll have a matrix that's, however many cells are in your data set, how many barcodes you had, along with about 20,000 or so different gene features. And so from this matrix, you can use dimensionality reduction techniques such as principal components analysis or TMAP or TISNE or UMAP in order to collapse all these features down to usually 2 vectors in order to visualize on a Cartesian coordinate system. Then once you have the kind of location of these cells, usually you use a marker-based strategy where you can say, oh, this cell had a high expression of CD4 and CD8 and CD3, therefore it's probably a T cell. Any of these manual kind of ways of doing it, or you can use a cell annotation software such as Castle or Cell Typist, which will use kind of a reference single-cell RNA sequencing data set in order to match your data set with what they have in order to see, okay, this is probably a fibroblast because its transcriptomic profile is similar to the reference the reference transcriptome. And so that's how you can get the cell types attributed to each one of these profiles. And feel free to interrupt if anybody has any questions. I know. Yes?
In the sorting part, how does it know cells?
So that's what I was just referring to. So that's you can use a manual marker-based approach. You know, if you have a high CD3, it's more likely to be a T cell, right? Or you can use a cell annotation tool. And these will basically...
During the initial formation of the droplet. Go back. Yeah.
Oh, sure. I mean the technology itself.
Yeah. Like, so when the barcode goes in, how does it know which cell to compare to?
So all of the transcripts from that cell will, well, not all of them, but hopefully a good portion, will bind to the bead, right, with the poly A tail. And so these have a 10x barcode attached to them. So all of those tags on this bead have the same barcode. And if you just have one cell within this bubble, or droplet, then you should have all of the transcripts having that same barcode. And so you know that belonged to that cell. Does that make sense?
You don't know what cell it is.
What do you mean, which cell?
Like, so you have a mixture of cells that go
Yeah.
in, and then it binds with the bead.
Yeah.
How do you know which cell type it is?
You're going to have to do that algorithmically from a program like this. Yeah. Yeah. So it's not perfect, but it's the best we have so far, right? So once you have your predicted cell type, along with the transcriptome, now we can start running cell-cell communication algorithms. So one of the earliest ones to come onto the scene was cell phone DB. This introduced the cell phone DB database in order to kind of manually curate a bunch of protein-protein interactions and other ligand-receptor pairs to kind of match your different signals with. This is version 1. They're up to version 5 now, I believe. And it has thousands of different protein-protein interactions and thousands of secreted and membrane proteins, et cetera, et cetera. Mostly we focus on this ligand-receptor pairs within the cell-cell communication field just because it's more, I guess, straightforward to do at this stage. But the concepts are very similar of how you would detect these throughout. So what does this database look like? Well, it comes in form of several different spreadsheets. So for example, here's the interaction spreadsheet where it will have the numerical or string-based ID of that interaction. It will have the sender molecule and the receiver molecule along with these are, again, IDs. Along with the source, so what data set it came from, UniProt, or maybe it came from some publication. It said whether it was manually curated or not, whether it's protein-protein interaction. It also will say what kind of communication it is, for example, the adhesion by collagen. To find out more information about the sender and receiver molecules, there's this multi-data spreadsheet. These are much longer. I'm cutting it off, by the way. I don't have the ... but these are much longer, obviously. And so you have the numerical and string names for the each molecule, along with whether it is a receptor or not, or a ligand, and whether it's secreted or it's a transmembrane protein or peripheral or integrin. So you can find out more information about each one of these candidate molecules from these kinds of spreadsheets. So with these databases, you now go through your algorithm in order to identify whether communication is occurring. So cell phone DB came with kind of a simple one to start out with. You essentially have your clustered single-cell RNA sequencing data along with your database of known ligand receptor pairs that are known to bind together. And essentially what they did was they looked at the average expression from a candidate sender cluster or cell type and see if another cluster has a high expression of the cognate receptor gene expression. If so, then they kind of have this candidate, cluster 1 is talking to cluster 2 with these, this mean value. To see whether it's statistically significant or not, they basically do a permutation test. Well, they will randomly shuffle the cluster assignments to each one of these cells and see whether their observed mean is larger than what they would get from that shuffle. And if that null distribution of the shuffle is much, much smaller than the observed, then we know that that's probably going to have a very low p-value. It's probably statistically significant that these two clusters are communicating with that particular ligand receptor pair. There's a lot of customizability for this kind of process. For example, within CellPhoneDB, they only consider ligands and receptors that are present in greater than 10% of the cells within that cluster. They randomly permuted the cluster labels 1,000 times. I would recommend maybe doing a bit more, just because permutation tests require many permutations in order to be reliable. They calculate the proportions of means as more extreme than what you would observe. And if there is a protein dimer, just like they have here, that's A and B, they want to look at the minimum gene expression, because you can't have kind of a lot from one of the dimers and half from the other and expect there to be more than the smaller of the two. Then with kind of these ligands and receptor pairs identified, you can visualize it in a variety of different ways. Here you can use something like Python iGraph, for example, to show that the CD4 cluster is talking to the dendritic cell cluster. The darker the edge, the more ligand receptor pairs were found to be communicating between these two populations. Okay, so this is kind of the general kind of overview of many of these types of algorithms to get started. So again, you just as in CellPhoneDB, you collect your single cell RNA sequencing or what have you from your sample. You have this in a matrix format for your genes within cells. You combine this with your ligand receptor pair database into these candidate pairs. You have some kind of scoring function with the ligand and receptor expression as input in order to see if they're really communicating or not, and then you visualize it in some way. So a lot of it is just like variations on this kind of scheme. So a few different kinds of scoring functions that you can use are, for example, expression thresholding. So this is kind of what CellPhoneDB used, where they said, oh, greater than 10% must be considered, right? So this is similar, where you're saying if the ligand expression in my distribution is high enough, and if the receptor expression, the cognate receptor expression, gene expression is high enough, then these are communicating, right? That's a 1 or a 0, is communicating or not. But you could also have an expression product by just multiplying these values together. This is actually kind of nice because it controls for cases where you can have an extremely high receptor gene expression, but if you have 0 for your ligand expression, then your overall expression product will be 0, right? It's not communicating at all. So that's nicely controlling for that. So this is a continuous function. You can have, similarly, an expression correlation where you have increased ligand expression correlated with increased receptor expression, similar to the expression product, which is also continuous. You can also do kind of this differential expression, right? This fold change between the ligand and receptor samples or cell clusters. What's nice about this is you also get a p-value associated with it. So instead of just having a single cutoff of saying, oh, it's this highly expressed or this highly differentially expressed, you can now also cut off based on what is significant or not, right? But this is binary. So as you can see, we can start to increase the space of the kinds of ways to interpret cell-cell communication. For a very extremely different way of doing things, for example, you can replace not only the ligand receptor pair scoring, but also the underlying model of communication itself. For example, this is what CellChat does. It uses the law of mass action in order to model not only the ligand-receptor pair expression, but also take into account co-inhibitory and co-stimulatory receptors and agonists and antagonists. And again, you'll see this very common theme where you get the ligand-receptor pair database. You pass that through to your data where you get the cell groups like those clusters, either through clustering or through the latent space that you cluster on top of, and you get those overexpressed genes per cell group. And then this is where they have like this new kind of model. So for example, they use their law of mass action, which is based essentially providing the value for the dissociation constant as part of a Hill equation. So they use Hill equations all over the place in their model. The Hill equation is basically the definition of the fraction of receptor protein that's bound by ligand. And this is defined as that ligand concentration raised to a Hill coefficient. This Hill coefficient will vary. If it's less than 1, basically that means that when the ligand is bound to a receptor, it is less likely to bind to more ligands. A receptor is less likely to bind to more ligands, and vice versa. If it's greater than 1, that means that if it's bound by a ligand, it's more likely to be bound by more ligands. CellChat just sets this to 1, so I'm not going to be really talking about it. That just means it's kind of independent of how many ligands receptors are bound. So basically, it's defining the ligand concentration divided by the ligand concentration plus dissociation constant from the law of mass action, which is basically what is the receptor at half binding concentration. So in CellChat, they define this as 0.5. The reason why is they normalize all of their gene expression, concentrations to be between 0 and 1. So half binding would be 0.5. So with that in mind, again, this is that fraction of receptor protein bound by ligand. They use Hill equations all over their model. So basically, the probability that a sender cell type I is talking to a sender cell type J with ligand and receptor pairs of L and R are defined first by this Hill equation of the ligand receptor the average ligand receptors multiplied gene expression multiplied by the average receptor gene expression on the receiver cell type. And these are defined not just by the expression, but it's the geometric mean of M1 subunits of the ligand, just in case there are additional subunits. Same with the receptor, is the M2 subunits. But this one is also modulated by the average of co-stimulatory and co-inhibitory receptors. Okay, so that's this component. That's just kind of like the ligand and receptor expression multiplied together, analogous to that. The second component is they're just multiplying it by modulating with agonists. So they have the Hill equation now for agonists from sender I or receiver J. And then the second part is modulating with antagonists from sender I and receiver J. But you'll notice the difference is that the antagonists have the dissociation constant on top, right? This is for the dissociation. This is where you have the competition for binding that the antagonists are blocking. And lastly, they weigh this whole equation by the numbers of cells belonging to each cell type. So Ni is the number of cells for sender cell I and sender cell type I, and Nj is the number of cells for receiver cell type J, divided by the total number of cells within the data set squared. So this is kind of how CellChat does it to have a more, they say, robust way of modeling the probability of these communication scores. Yes?
Does this take into account the distance from one cell to another, or just
no
specific? Okay.
No, and that's important for the second half of this talk. But yes, so that's a huge problem. which can potentially lead to a lot of false negatives and positives. But yes. Oh, but Cell Chat version 2, I believe, is on bioRxiv, and they're purporting to take that into account. And I think it's through thresholding. I think they say, oh, if you're, don't quote me on this, but I think it's based off of a threshold of distance, and then they have a similar model. But yeah, so this is the original one from 2021, 2024, or whatever. The bioRxiv, I don't think it's published yet. They're trying to take that into account as well. Yeah.
So all of this is based on like cluster of cells, so
yes,
basically like identical cell types or similar cell types. What happens in cases where you have like a subset of cell type A talking to a subset of cell type B? Is that just completely missed in?
Yes, you'd have to do subclustering in order to find that out. However, there are very, very, very few algorithms that try single cell, including ours, based communication. But the vast majority that you'll see will do cluster. Yeah.
The amazing talk, thank you. The spatial would be a solution for that, right? Spatial transcriptomics.
Yes. What about spatial transcriptomics?
And then you could that would solve the problem how close the cells are to each other.
Yes, exactly. I'm going to go over that too. Yeah. Yes. So there's many kinds of methods that do the cell-cell communication. There's cell phone DB, cell chat. Connectome has one, single cell signal R, scribe-in, NatMe. There's many out there. So actually one of the other algorithms called Liana attempts to kind of create a consensus of all these communication algorithms. So they'll take, it takes many different databases that were identified along with, you gotta be careful though, some of these are repeats, like OmniPath is also trying to collect a lot of these data sets as well, but I'm sure it's all normalized and and controlled for. And then it takes a whole bunch of different methods, and it finds a consensus along with your clustered single-cell RNA-seq data to identify a more robust cell-cell communication mapping. Okay.